Now we know lots and lots about DNA. We know its structure, we know what it looks like, we know how it works, how it codes for your genes, which are codes for specific proteins, which make you, you. We know how to copy it and make more, so when your cells make more cells, we can all get copies of it in each and every cell. So now I want to take just a little bit of time to talk about what do we use all this stuff for in the real world. Because I know a lot of times in high school we forget to make that connection because we forget that you guys can't do that on your own. So DNA is really important because it is who we are. Every single person, except for identical twins, has a unique genetic code different from everybody else in the entire world because we all have a different combination of A's, T's, G's, and C's. We're all made out of A's, T's, G's, and C's, but each and every one of us, it's a little bit different and a little bit different of an order and grouping. So we can use all this information to our advantage. Let's talk about a couple of ways. One thing that was a big, big project about a decade ago, a little bit over a decade ago, was the Human Genome Project. The Human Genome Project was an effort of scientists all over the world working together to map out the entire sequence of the human genome. So every single A, T, G, and C in a human. And it took 20 years to be able to do it the first time. But we learned so much in that process. One thing we learned was how to do it better. So now we can do it faster. Now it only takes a couple of months. The first time it took 20 years. Now it just takes a couple of months. And it's a lot cheaper too because it costed several $billions to do the first one. But we were hoping that we could detect new genes. We could detect ways that genes have gone wrong in diseases, hopefully prevent some diseases, and hopefully find treatments or cures from genetic diseases caused by problems in a human's genome. And we discovered a lot. We did discover a lot of new genes. One gene that was discovered was a gene that's linked to breast cancer in women. If you have this gene, you have a 90% chance of developing cancer. So now women can go in and they can be tested for the gene so that they can determine what they want to do about it if they test positive. Do they want to just change their lifestyle and get more regular doctor visits to detect it early, or do something radical and go ahead and have a full mastectomy and get them completely removed so that you can't get breast cancer if you don't have breast tissue? There was a really famous person who did that, Angelina Jolie, who found out she had that gene, so she went ahead and had a mastectomy and had them completely removed, so now she doesn't have to worry about developing breast cancer. We were also hoping that we could one day do what's called gene therapy. Gene therapy is the idea that you can find a gene that is defected, remove it, and replace it with a correct copy of the gene so that now it will work correctly. This would be a cure for all kinds of diseases that are linked to a specific gene or a set of genes. There's still a lot of trouble with this. We're still not really good at it, but there's lots and lots and lots of current research going on in this field. We use DNA technologies all the time to help us identify, study, and modify genes. One way that we use DNA that I'm sure you all know about is forensic identification. Being able to find out who was at a crime scene, who left blood somewhere, who the identity of somebody is if their body is so badly decomposed that you can't tell by looking at them. You can use DNA and fingerprints and things like that to identify them. One thing that you guys need to be able to do is know how to read a gel electrophoresis. So a gel electrophoresis, we use this to determine who the baby daddy is. So Jerry Springer uses this all the time. Or what suspect left blood at a crime scene. So what you do with the gel electrophoresis is you take a sample of DNA and you do something called PCR, which replicates the DNA a whole bunch of times. We know about the process of replication, so we're going to replicate it a whole bunch of times so that we have a big sample of DNA. Then we're going to cut the DNA with restriction enzymes. We've talked about those before. Restriction enzymes are special enzymes that just cut the DNA in certain places. Well, since everybody's DNA is unique, then everybody's DNA is going to be cut in different places. And so you're going to cut the DNA up, and then you're going to run it in a gel. And that's going to be our lab activity that we're going to do in just a little bit, is we're going to run a gel. So you can see how it works. So if we look at this sample here, we have a blood stain in this first column. This was found at the crime scene. Okay, we've already checked. It doesn't belong to the murdered victim. So that must mean it must belong to the culprit of the murder, the murderer. So police canvassed the neighborhood and interviewed possible witnesses and came up with 4 possible suspects: Bob, Sue, John, and Lisa. So the cops went and got a warrant from the judge to obtain samples of DNA from these 4 people. Getting a sample of DNA for something like this is very easy. It can just be with a cheek swab, because when you swab your cheeks, you get cells that come off, and all cells have the DNA, so you can do this sample. Okay, so they took blood samples from all these 4 people, cut them with the same restriction enzymes, so everybody got cut with the same restriction enzymes, but since everybody's DNA is unique, it all gets cut differently. So take a moment to look at this and decide which one of these suspects has some explaining to do, because their blood is the blood at the crime scene. Hopefully you notice and you picked John. John has some explaining to do because his DNA was cut identical to the blood at the crime scene. 2 bars here, 2 bars here. So this means that this blood is his blood. His blood was at the crime scene. So he is now our prime suspect, and he's got some explaining to do. We've already learned about another DNA technology advancement that we can do, known as bacterial transformation. This is where you take a plasmid, okay, of DNA. You can put in a human gene, such as the gene for human insulin, put it inside bacteria, and cause the bacteria to create human insulin for us. Okay, that whole process, the way that we make insulin, is called bacterial transformation. With the whole activity on it. So if you don't remember, you can go back and still watch that video on bacterial transformation, plasmids, and making insulin. Something we can now do is called genetic engineering. In fact, making insulin is an example of genetic engineering. To do genetic engineering, we're going to use what's called recombinant DNA. This is where you take DNA from one species and put it in the DNA of another species. So we're going to take one gene and we're going to put it into another species so that they can use that gene. That's what we do with the human insulin gene and putting it into bacteria. It's using recombinant DNA. But we can do this on a larger scale now. And you guys have heard of this stuff because they're called GMOs, genetically modified organisms. And you've got some radical groups who are uneducated and don't know what GMOs really are, saying that they're bad for you, they're bad for you, they're going to kill you, they produce chemicals that are bad for you, which just simply isn't true. GMOs are actually really, really good. And GMOs have been around since the beginning of farming, because when a farmer used to take, let's say he's growing corn, and he takes his biggest ears of corn and he saves them and uses them to start his crop next year, well, he's genetically modifying his crops by choosing the best and allowing them to be the ones to create the next crop. And then that year, he chooses his biggest and best ones and saves them for the next year's crops. That's genetic modification at the most basic level. And so pretty much all major food sources now are genetically modified like that. Like, think about if you've ever seen a wild strawberry. Wild strawberries are teeny tiny. They're about the size of your pinky fingernail. But humans have genetically modified strawberries by choosing the the plants that produce the biggest strawberries to mate and produce the next season. And so now we have gigantic strawberries the size of your whole hand, because, well, we chose to breed them that way. And that is, in its simplest form, a form of genetic modification. Now we can actually go in and manipulate the DNA by taking genes from one species and putting in another. For example, our dairy cows are genetically modified. We took a gene from another species that isn't good for producing a ton of milk for us, because they only produce a little bit of milk, and we put that gene into our dairy cows. And this gene makes them resistant to udder infections. And that's a good thing. It's good for the cow, because now they're not going to get these udder infections, which are incredibly painful and cause a lot of pain and bleeding from their udders. And it's good for the farmer, because if you have a cow with an udder infection, any milk produced by that cow has to be thrown out. It can't be used because it has bacteria in it. It's infected milk. It's garbage. But now that our cows don't get udder infections as often, the cows are happier because they don't have udder infections and bleeding udders, and the farmer's happy because they're producing more milk that is actually sellable. Seedless watermelons. Seedless watermelons are not normal. They are genetically modified. We modified and took out the gene that tells them how to produce their seeds, so they don't produce seeds. If they don't produce seeds, they can't produce more watermelons, right? So they can't reproduce on their own. And there's lots of examples of this. Ears of corn are huge now, and they're resistant to pests eating on them because we've genetically modified them. We've given them a chemical from another plant, and the chemical that they produce, that gene that lets them produce that chemical, only goes into the leaves of the corn, and we don't eat the leaves in the stalk of the corn. So we're not eating that extra chemical that's being created, because that's not the part of the corn that we eat. But it makes the leaves of the corn taste really, really bad to insects, and can even kill some insects. Insects don't eat them and don't harm them, and so we have better corn crops now. What I think is really cool that we can do is we can now grow certain body parts on animals. So these little mice, like you see here, they have zero immune system. They've been genetically modified so that they don't have an immune system, so that we can get them to grow a human ear or a human nose on them. And these little mice live really good lives, actually. They, have their nice own little cages. They get fed every single day. When they have surgery, to have the scaffolding for the ear placed on them, they're put to sleep just like humans, so they don't feel any pain. They're given painkillers, and they just run around and they play all day while they grow their little ear. And then after they've grown the ear, they have surgery again, but again, they're put to sleep so they don't feel any pain, and they're given pain medication while they heal. And after they've had a surgery, they get put out to pasture, meaning for the rest of their little mousy lives, they get to play, feed, and reproduce to make more of these little mice for more surgeries. So they only have to go through the surgery process once, and then they live out their little mousy lives, eating, sleeping, and sexing. So pretty nice little life for the mouse there, right? Okay. So I think this is pretty cool. There are some animal rights activists who say that this is wrong. And there was once a group that they broke in and released thousands of these little mice. Well, let's think about that. These mice have zero immune system. That means any bacteria they come into contact with is going to make them incredibly sick. So when that group released these mice, those mice all died horrible deaths from horrible diseases. They began vomiting, they had terrible diarrhea, and they died horrible little deaths. And if this animal rights activist group had educated themselves and known that these mice actually have a really good life and they actually killed them in a horrendous way, maybe they wouldn't have done that. I'm all for animal rights activists, but if you're going to do it, be educated. Go after the people who are actually harming the animals and not following laws. There are laws in place to protect animals' rights, and most research facilities follow them.